Where is she? Brianna, you might be the only one person, student coming in. Can you hear me?
Yes, I can hear you.
Oh, well, let me turn my sound up because I can barely hear you. Okay, I'll wait a minute or so and see if anybody else comes in. Did you have trouble? Did you have trouble getting in?
So I actually went on to D2L and went on the meeting links, and I was on that. But I'm not sure if that one was different than the one that you emailed me, but the one that you emailed to me worked perfectly fine.
Yeah, I think I may have to reset uh, those links that are in D2L. I'm not sure they're working. But I think when you clicked on it, I got a message saying that you joined the meeting, but you weren't in the waiting room, so that's why I sent you the invite. But I'm hoping if anybody else tries to get in, I'll get an email and then I'll send them an invite. All right. So anyway, um,
Are we going over the genetic problems that you posted? It's like a whole bunch of them.
yeah. The one that we should go over is part 2 genetics problems. I just realized that the other one that I sent was almost identical. So, well, let me pull it up. I got it. I can screen share here and show you. This is what I'm talking about. Where is it? Yeah, this one right here.
Okay.
So you get a copy of that.
Yeah, I have it pulled up as well.
Okay, so you know how to do the regular Mendelian problems with a Punnett square.
Yeah, I believe so. Just a little rough on it.
Okay, well, we'll look at the first one. Mid-digital hair and knuckles between the first, second knuckles, a normal dominant human trait. Cross two people who are heterozygous for allele, what is the chance that their offspring will express this trait? Did you try that one?
No, I haven't honestly tried any of them because I knew that we would be going over it, so I just wanted to be 100%
Okay,
on how to do it.
okay, so the first thing, well, if I asked you, well, on the exam, I'm not gonna, you won't be drawing Punnett squares on exam, but you should have some pencil and paper in front of you. It's an open book, open note exam, and it'll give you like this problem right here. It'll give you this problem, and then it'll give you a multiple choice. So what is the chance that their offspring will express this trait? And you'll have 100%, 75, 50, 25, and 0. So it works out. So it works out well for a multiple choice question with 5 choices from 100 to 0. And so you have to do this and perhaps sit down with a pencil and paper in front of you and actually do the Punnett square to see which is the correct percentage. So when you do that, remember, do a Mendelian problem like this, you select a letter for the dominant allele and the same letter for the recessive. Don't pick a different letter. So for example, mid-digital hair in the knuckles, let's pick E. Well, you can pick any letter. K is a bad letter to pick because you want to be sure know the difference between the big letter, uppercase letter, and a lowercase letter, and they look about the same. So if you pick an E, so big E would be hairy knuckles. Small e would be not, right? So two parents who are heterozygous for the allele, they will both be big E, small e, right?
Yeah, so would you put big E, small e at the top, and then on the side you would also put big E, little e going
That's
down
correct.
the side? Okay.
And so what is the chance that the offspring will, why don't you go ahead and do that? Tell me what's the chance that the offspring will express this trait?
So what is it, which, um, hold on. Okay, sorry, I just need to reread it.
Okay.
Would it be 50%
No, it wouldn't be.
or 25?
No, remember, this is a dominant trait.
See, this stuff confuses me.
Yeah, you got to be careful. What are we talking about? A dominant trait or recessive trait? This is a dominant trait. So that means all you need is one big letter and you will express it. So if you cross big E small e versus big E small e, what percent of there's 4 possibilities.
So it'd be 75
It'd be 75
because
because
there's
three of them have a big E. There's
big
only
E.
one.
Okay.
Right, so that'd be 75%. Now, if you go down to 3, albinism is a complete lack of development. Now, you also said albinism is a recessive trait. So here are 2 people who are heterozygous for albinism. So it's the same cross, but this time it's a recessive trait. So it's big A, small a versus big A, small a. Now, what % of the children will be expected to be albino?
50.
No. Now remember, it's a recessive trait, so you need 2 small letters to express it.
So 25.
25%. There's only 1 out of 4 is 2 small letters. So so it's, you do the same thing each time for these Mendelian, these monohybrid crosses. Just make sure you know whether it's a dominant or recessive trait. Okay.
Okay.
All right. Number 2. Among Hereford cows, a dominant allele called pole. This one lacks horns. You're a cattle bear, and you have you make sure each cow has no horn. But among the calves that year, some grow horns. You dispose of those, and so that no horned adults got into your pasture. Next year, however, more horned calves are born. What's the problem here?
This one is tricky.
Well, so there's a dominant allele called polled. So say that's big P. And you don't have any, and all of your whole herd doesn't have any horns, but you mate a bull with a cow, and some calves have horns. So why is that? So remember, having horns is a recessive trait, right?
Yeah.
So so you have to have 2 small letters. So what's happening is this, recessive trait can be hidden. So so the problem is that the parents are heterozygous for it. They're carriers for horns. So expect 25% of their offspring would have horns. So what you want to do is try to get rid of that small letter among your cattle, so you don't have any horns showing up at all. And it's particularly dangerous with the bulls, because you use bull sperm to inseminate all of the cows. So how are you going to find out whether so you don't want to be mating a bull that has a small letter that's heterozygous for this. You only want to have bulls that have 2 big letters. So how are you going to find out which bulls have 2 big letters and which bulls have a small letter? Because what the problem is, they look the same from the outside. So the homozygous dominant phenotype is the same as the heterozygous phenotype, which in this case is no horns. So how are you going to find out whether the bull that you're using to mate all these cows is heterozygous or homozygous dominant?
Would it be to reproduce or no?
Yes, to do a cross, but with what? So you take the sperm from the bull, and which cow do you inseminate?
The one with the No horns?
No, because you can't tell from the outside whether an individual without horns is homozygous dominant or heterozygous. You will mate you will mate that bull with a cow that you know just by looking at it what
Okay.
its genotype is, and that would be a horned cow with
Okay.
horns, because you know that cow has two small letters, right?
All right.
Okay, so now you make that bull, and if you get any calves with horns, then you know that bull has a small letter, because the only way you can have horns is having two small letters. If you make that bull with a lot of cows and there's not one calf with horns, then you can be pretty sure that that bull is probably homozygous dominant, and you can use his sperm and not have worried about getting any calves with horns. See what I mean? Because if
Yeah.
so this is called a test cross. Right? So you gotta know how to do a test cross, and when you do a test cross.
Okay, this was on the one lab, right? The test cross. It just asked
Oh
like a
yeah,
couple
yeah. As a
questions.
matter of fact, it was on the genetic simulations. There was a test cross that you and it will be if you do a test cross, a heterozygote versus a homozygous dominant, you expect half the offspring will express the recessive trait. So so if you make this bull with several cows, there's a good chance that if he's heterozygous, he's going to have a calf with horns, and then you know that bull is heterozygous.
I'm just gonna make sure that I really go back in the textbook or the PowerPoints and
Yeah, and
make
go.
sure I get to know this because it still confuses me. Like I understand what you're saying. It's just hard to process like really fast how to do it.
Yeah, yeah. So you got look over. I mean, this is in the. Let me see in the book here. Well, let's hope it's in the book. Let me look this up here. It's a test cross. Well, test cross is in the PowerPoint, but I'm going to see. I just tore the page. What the? Okay. Test cross. Yeah, test crosses 152. Yeah, so it's
And pro
on
152.
page 152. Is using a test cross to determine an unknown genotype.
Okay. And like these types of problems will be on the exam.
Yes.
Okay.
Well, that asking you, you can't tell the difference just by looking at an individual, whether they're homozygous dominant or heterozygous. They have the same phenotype. So you have to do a test cross in order to determine that. Okay. Okay. So we got test crosses. Look at number 4. So sometimes, in these problems, you'll be given the genotypes, and then you just do the Punnett square. Oh, somebody else wants to come in. Hang on a second. Okay, hang on a second. I'm going to send her an invite.
Okay.
Oh, wait a minute. What's, hang on a second. Carly with a C, not with a K. Okay. Okay. Number four. So you might be just given the genotypes and do the Punnett square. Otherwise, some problems, you'll be told a situation. You got to figure out the genotype from the information in the problem, like a number 4.
So for this one, we would just do a Punnett square.
Yeah, for number 4, you just do a Punnett square, but it says that so a dominant condition for earlobes is attached, right?
Yeah.
So a woman has the attached condition is heterozygous for it. So let's say you can use big L and small l. So she is big L, small l. Her husband has unattached ears. So what is his genotype?
So would it be big L, little L on the top, and then 2 small L's on the side?
Right, 2 small l's because it's
Perfect.
her
He
husband
doesn't
has
have the, he doesn't
unattached
have
ears,
the,
and that's a
yeah.
recessive condition, so you know it's got to be 2 small letters.
Okay.
So what is the chance that each of your children will have attached ears?
50%?
50%. It's 50-50. It's like a test cross. Heterozygote versus a homozygous recessive.
Perfect. I'm glad I understand those ones.
That's good. That's good. So then we got to get into, going to the next page. Number 5 is the same as the one with the cattle. You have a Drosophila, and you want to know whether your fly is heterozygous for, oh, let me let Carly in. Hang on a second. You want to know whether your fly is heterozygous for a recessive allele, so you got to do another test cross. That's another test cross question.
Yeah, I'm gonna make sure that I just really go over the test cross because I understand the Punnett square questions, but the test crosses are the ones that I get confused.
Well, remember, it's just that, as I said, the an individual that's homozygous dominant and a heterozygous, and if they have the same phenotype, so you can't tell from the outside what their genotype is. You can't tell.
Right.
So you have to mate that individual with a homozygous recessive individual and look at the offspring and see if any of the offspring have the recessive trait. Okay. So Carla, you can hear everything.
Yes.
Okay, good. You got any questions? We're just going over the these problems that I put up on D2L.
Okay.
Now, number 6. Sometimes you'll be given data, and you got to figure out the nature of the crooked beak allele. So you pigeon breeder, you get two a male and a female pigeon. They're completely normal, but and they produce 23 offspring, but 5 of the offspring have a crooked beak. So what's the when they say what is the probable nature of the crooked beak? Is crooked beak going to be dominant or recessive?
Which paper are we on? Are we
Well,
on?
we're on page 6 of part 2 Gen X problems, page 6.
Okay.
Oh yeah, let me pull it up. Hang on. Share screen again. Okay, let me go down here. Yeah, this is page 6 right here. This one right here with the pigeons. Brandon, are you still there?
Yeah, I'm just not sure about this one.
Okay. Oh, okay. So here's the thing. If you have two parents who don't show this trait, but they have an offspring that does share the trait, it has to be recessive. It can't possibly be dominant. And
So
go ahead.
the genotypes of the parents, it would be heterogeneous with like an uppercase letter and a lowercase letter.
Well,
Because
heterozygous.
yeah.
Yeah, they'd have to be heterozygous um, because this is a recessive trait. As I said, because if it were a like I said, it can't possibly be dominant, because if it was dominant, then the normal would be recessive, right? And then two normal parents would be two small letters, and they can't possibly have a baby with a big letter, because they haven't got a big letter guy. So if the parents don't express it, and it ends up and it's shown, but they have a offspring that shows it, it has to be recessive. Okay, and the genotypes of parents have to be heterozygous, because they both have to give a small letter, so they have to be heterozygous. And you expect them, about 25% of them, to have the crooked beak, because two heterozygotes making babies, 25% of the babies are going to have two small letters. And that's about 25%. 5 out of 23 is about 25%. Okay, so we see that. Okay. Now if you go on to 7, it's the same situation. Remember, when it says two parents are carriers, remember what a carrier is. A carrier is a heterozygous individual that's carrying a recessive allele, and therefore doesn't express it, but can pass it on to their offspring. That's a carrier. So big C is normal, small c is cystic fibrosis. Then both parents are big C, small c. And here they're not asking what percent of the offspring will have cystic fibrosis, which would be 25%. But how many carriers will they have? So they are carrying how many carrier offspring? And that's going to be 50%. Remember, you do put a square, upper left, big C, upper right, big C, small c, lower left, big C, small c, lower right, small c, So that's 2 out of 4 are heterozygous, and so that the answer to that is 50%. Do you see that?
Yeah.
Okay. Now, number 8. So it's hypercholesterolemia. More cholesterol, so it's controlled by a dominant allele. So a heterozygous, well, it shouldn't, actually, that's redundant. If a heterozygous carrier, if a carrier marries a homozygous individual who does not carry the allele, so that would mean 2 small letters. So this is again, it's like a test cross. It's a heterozygous individual making babies with a homozygous recessive. So what's the chance of the children carrying the allele? And that'll be 50%. Half of them are going to be carrying, because the heterozygous parent is going to give, on the average, half of his kids the big letter. What is the chance that a child will have the condition? That's also 50%, because half of them are going to have the big letter. So are we clear on these?
I think so.
Now, oh yeah. Okay, I guess we should talk about, and also when we stick with Mendelian, the next one is a non-Mendelian. So let's switch to number 14 on page 8, to a dihybrid cross. So we got to be able to look at a dihybrid cross. Oh wait, first 12. So like I said, you won't be having to do a dihybrid cross on the exam, but you should recognize how you get the allele combinations for dihybrid cross. And also, if you look at the results of a dihybrid cross, what does that mean? And looking for number 12, remember a dihybrid cross has 4 letters, right? Not 2. And so this is dumpy wing is recessive small d, normal wing big D. And then red eyes are normal, are we call wild type, they're dominant. White eye is recessive. So in a cross of big D, big W, small w, times big D, small w, small w, what proportion of the offspring is expected to be normal, that is long wing red eye? What proportion is expected to be dumpy white? All right. So here's the thing. What is the genotype of a normal individual? Well, first look at the wings. If you have normal wings,
Big D.
What, excuse me?
Big D.
Big D, and something else, And it doesn't matter what that other thing is. If you have one big D, you have normal wings. So big D, big D, small d, both have normal wings. Okay. Same thing with the eyes. What is the genotype of a red-eyed fly? It's big W and something else. So the genotype of a normal fly with normal wings and red eye is big D something, big W something. So any of the offspring that has one big D and one big W is going to be normal, completely normal, right? What would be the genotype of a dumpy white? Dumpy wings, white eyes.
Little d,
Right?
and big W, and then something.
No, it wouldn't be big W.
Or sorry, no, I was thinking red. It would be little W.
Right, so it'd be all small letters. That's the only individual that's going to be dumpy white, all small letters. Okay, to do the Punnett square, you'd have to find the allele combinations. So big D, wait a minute, can you see? So this is an extra step you have to do in dihybrid crosses. You have to get the allele combinations. That is what alleles are going to be in this parent's gametes. So big D, big W, small w can make, let's say it's the male, can make sperm with a big D and a big W, and can make sperm with a big D and a small w. So that's it. Do you see how I'm getting at?
Yeah.
So this is an extra step you have to do with dihybrid crosses, because in a monohybrid cross, you just take the two alleles and you put them on a Punnett square. But in dihybrid crosses, there's going to be two alleles in each gamete. So you got to find out what two alleles. So it's only big D, big W, big D, small w. Okay. The other parent, say the female, big D, small w, small w. So what gametes can she make? What are going to be in her gametes? Well, she's going to give one of the D's to her offspring, right?
Yeah.
So she can give a big D or a small d. What about the W site? You can only give a what?
Could you repeat the question?
Okay, so what alleles are going to be in her eggs? It's going to be, she can make eggs with a big D, right? She can also make eggs with a small d. But what's going to be in the eggs, what allele is going to be in their eggs for the W site?
Big W, right?
No, she doesn't have a big W. She only has two small
So
W's.
confused.
So remember, you take one allele from the D site.
Okay, I understand. I was looking at something.
And one allele from the W site. She has two small W's, so she can only give a small W to her offspring. See?
I see that now.
Okay, so her allele combinations are big D, small w, and small d, small w. So this is a 2x2 Punnett square. It's not a 4x4, Punnett square like in the book. So what you do is you take you do a Punnett square, but you remember, make sure you put the two letters. So you got big D, small w, small d, small w on top of the Punnett square. And then on the side of the Punnett square, you have big D, big W, big D, small w. See what I mean?
Yeah.
Okay. So then you drop down and you bring across just like a regular, Punnett square. So top right, you're gonna have big D, big W, small w. Top right, big D, big W, small w. Lower left, two big D's, two small w's. And then lower right, big D, small w, small w. So you get what I'm doing?
Yes.
So the question is, what proportion of offspring are expected to be normal? That is, have at least one big D and at least one big W.
Is it 50%?
50%. The two top left and top right, they both have a big D and a small W, and the ones below have a small W. They don't have a big W, so they wouldn't be completely normal. They would have normal wings, but white eye. So what percent expect to be dumpy white? That is all small letters.
So would the D have to be lowercase as well?
Yes, for dumpy white, you have to be small d, small w, small
So
w.
would it be 0%?
It'd be 0%. There's no chance. Okay, so like I said, a dihybrid cross doesn't always have to be a 4x4. It could be a 2x4, it could be a 4x4. It could be a 2x2 or 2x4 or 4x4. Okay, one other thing about dihybrid cross is down on number 14. So marigolds, yellow flower dominant over orange, normal leaves dominant over mottled leaves. So you got a yellow flower plant with normal leaves, so in all big letters, and an orange flower plant with mottled leaves, all small letters. All of the offspring, the F1 generation, will be yellow with normal leaves and be double heterozygotes. All right. So now you made two double heterozygotes together, and this will be a 4x4, and you end up with 4 phenotypes: yellow normal, yellow mottled, orange normal, and orange mottled. Now the yellow normal is called a parental phenotype, and the orange mottled is a parental phenotype, because that was the cross in the P generation with a yellow flower plant with normal leaves, all big letters, and an orange flower plant with mottled leaves, all small letters. The offspring are all heterozygotes, and so then you cross two double heterozygotes, and you get what's these 4 phenotypes, 2 of which are called recombinant phenotypes, because he didn't see them in the parents. The yellow mottled and the orange normal. Okay, so what can you say about the location of these 2 genes? And don't worry about map units. We're not going to do anything about map units. Forget that part. But what can you say about the location of these 2 genes? Here's the thing you got to look for: the 9:3:1 ratio. You got to know what that means, and you got to be able to analyze a dihybrid cross and say whether this is 9:3:1 or not. Do you know what the 9:3:1 is?
I remember it was a question on the quiz.
Right. Here's the thing. So if you have a 4x4 cross with 2 double heterozygotes, you have 16 possibilities, 16 genotypes, right? 4x4 is 16. 4 times 4, right?
Yeah.
Okay. So then what you do is you count the number of individuals. How many individuals have that particular phenotype? The yellow normal phenotype. How many have the yellow mottled? How many have the orange normal? And how many have the orange mottled? You count those up. So yellow normal, and what you're looking for is independent assortment. Are these 2 loci independently assorting? That's one of the big things you want to find out about in a dihybrid cross. That's why Mendel did dihybrid cross. He wanted to see whether the segregation, the mixing of alleles at one locus has anything to do with the mixing of alleles at another locus. If they don't, that's independent assortment. So you'd expect 9 out of 16, or about 56%, would be yellow normal, because that would be a big, at least one big Y, at least one big N. 9 out of 16 have that. All right. Then the 3 is 3 out of 16 would be yellow mottled. That is a big Y and then 2 small N's. 3, so that's about 19%. And then 3 out of 16 would be orange normal, have two small Y's and at least 1 big N. And then orange mottled, all small letters, you expect that to be 1 out of 16, that's 6% would have that phenotype. So that would be 9:3:1. 56%, 19%, 19%, and 6%, 9:3:1. Okay, if that is true, then those 2 loci are independently assorting. And what that means is they are probably on different chromosomes. All right? You get that?
I think so.
So what you have to do is analyze the data and say, is that a 9:3:1 ratio? So you need a calculator for this. How many total offspring? There was 65.
I think it's 142,
Yeah, it's 142.
I believe.
Okay, so is this a 9:3:1 ratio? Well, let's see. 65 divided by 142 for the yellow normal is 46%. That's pretty close to what you expect. You expect to be 56%. It's 46%. So that's somewhat close. How about 3 divided by 142 is only 2%. Now you expect it to be close to 19% if it was independent assortment. And then 4 out of 42 is only going to be about the same. It's almost 3%. So they're way too low. It should be around 19%. And then you look at the orange model and you say, wow, is that off? That's supposed to be 6%. It's half. It's 50%. 70 out of 142. It's like half. So it's way, way off a 9331 ratio. So these two loci are not independently assorting. And what that means is that they are close together on the same chromosome. These are what are called linked genes on the same chromosome. This is something, and this is a non-Mendelian trait. Mendel didn't even think about this. So if it doesn't fit a 9331 ratio, you can say beyond a shadow of a doubt with 100% certainty, they are close together on the same chromosome. However, if they independently assort, you can only say that they are probably on different chromosomes. Because if they are really far apart on the same chromosome, they will independently assort. Because there'll be a 50% chance of a crossover between them. And that means they'll independently assort. So that's why you can only say they're probably on different chromosomes. And even with Mendel, two of his traits, all of his traits independently assorted, but two of the traits are actually in pea plants on the same chromosome. They're linked, but they're so far apart that they independently assort. So he thought all, traits independently assorted. They don't. They have to be on different chromosomes or really far apart on the same chromosome. So you'll get probably a question on independent assortment on the exam.
So for this question, it was not independently assorted?
Not independently assorted because it's way, way off a 9331. It's not even close to a 9331.
Okay.
Okay. Well, sticking with non-Mendelian now. So remember what Mendelian traits are. Complete dominance or complete recessive. That's complete dominance is a Mendelian trait. Independent assortment, Mendelian trait. Only two alleles possible at a locus. That's Mendelian. Right? And one gene controls one trait, one characteristic. That's also Mendelian. But if it's anything different, it's non-Mendelian. And here are some things that are non-Mendelian. For example, human blood type locus. Remember, I talked about this in the video lectures. The AB, your blood type, A, B, AB, or O. I'm not talking about the plus or negative. That's called the Rh factor, and that's actually a Mendelian site. Having the positive is dominant, having the negative is recessive. But at the human blood type locus has a couple of non-Mendelian traits. One is called multiple alleles. Where Mendelian traits only have two possible alleles at a locus. If there's more than that, then that's called multiple alleles. And the human blood type locus has multiple alleles. It has a allele A, B, and O. So there's three possibilities. So that's called multiple alleles. So if you see a problem on exam and there's more than two alleles possible, then you recognize that as multiple alleles. All right? That clear?
Yes.
Yeah.
Okay.
Can we go over question 9?
Sure. Okay. So Mary Lumina. So and here's the other thing about blood type locus. There's another non-Mendelian trait called codominance. That is, more than one allele is dominant. So in the human blood type locus, we have 3 possible alleles: big A, big B, and small I. And I is O. We'll give you O. So here's the possibilities of genotypes and phenotypes for the ABO locus. If you're blood type A, for example, I'm blood type A, so I have at least one big A. So I'm either big A, or big A, small I. Remember, I is recessive. Okay? Now, blood type B, you could be big B, or big B, I. Because I is recessive to B as well. See that? Okay. But since A and B are both dominant, if you get a big A from one parent and a big B from another parent, you have blood type AB, and they are both expressed. Now, what these things are proteins on your red blood cells. There's the A protein and there's the B protein. If you type A, you have the A protein on your red blood cells. If you type B, you have the B protein on your red blood cells. If you type AB, you have both of those proteins, right? And they're both completely expressed, co-dominant. And what is your genotype if you have blood type O?
Would it be the lowercase I?
I, 2 small i's. That's the only way you can be O, all right? Okay, married woman has blood type O. She has a child with blood type O. Her husband is blood type B. Is that possible?
If he was BI, right?
Right. So he could be, obviously, if he's the father, he is big B, small I. All right. Out of the blue, acquaintance of theirs claims he had an affair with a woman. He says he's the father of this child. His blood is tested and he's AB. Does he have a case, or is he just a lying scumbag?
It
Could
wouldn't
he be, excuse me?
It wouldn't be a case because isn't it dominant?
Well, yeah, the child is II. He is AB. Could he have an O child?
Probably not, because isn't the O recessive?
Right. And so he would have to have an I to give his child, because the child is II. He doesn't have a small I. So he can't possibly be the father of an O child. See? So that you say with 100% certainty. But is the husband definitely the father? Is the husband definitely the father?
He probably is, but I
He
guess
probably is. Yeah, he could be the father. But remember, but that's what paternity tests are for. Remember, mothers always know exactly who their children are, but fathers, we can't be absolutely sure. But he's probably, he could be the father. but the other guy can't possibly. Okay, everybody see that? All right. Okay. Next one. Sex linkage, another non-Mendelian trait. And what does sex linkage mean? Where is this locus located?
Isn't it is it the X chromosome?
X chromosome or the Y chromosome. Remember, there are two types of sex linkage, on the X or on the Y. Right? Now, but usually, if the problem just says sex linked, it always means X linked, because the Y chromosome is this tiny little chromosome and it doesn't care very much, and it's only carried by males, so it's not that interesting to look at. So if it just says sex linked, it means it's X linked. Okay. Okay. How do you set up now you have to set up a sex linked cross here to determine the answer to this. How many of the boys would be colorblind? How many of the girls would be colorblind? Okay, the way you do that is first you write down the sex chromosomes for mom and dad. So for mom, XX, right? For dad, XY, right? That's the first thing you do. And you pick a letter to represent the dominant allele and the small case letter for the for the recessive. So big R for normal vision, small R for colorblind. Okay? Got that?
Yes.
Okay. So this woman is colorblind. What is her genotype?
Wouldn't it be the two small R's?
Two small R's. Now you put those two small R's as superscripts above the two X's. So that's her.
How would you do that?
Well, you know what a superscript is.
Doesn't it kind of look like an exponent?
Yeah, it's like an exponent. It's so you have the X and you get the little R to the upper right, to the like you say X squared. You put X, but you put a 2. Well, here you put a letter,
little R's.
A small letter. So it's X small R, X small r. All right. So that's on the top of the Punnett square. Okay, what now? Her husband is not colorblind. What is his genotype?
Wouldn't it be a big N? And the Y wouldn't have a super.
Right, it'd be one big. Well, I'm using R, using N, doesn't matter. So it'd be X big R, and there's nothing on the Y because this is X linked. Okay. Now, how many of their boys will be colorblind? What percent? Go do Punnett square and tell me the percent.
Would it be 100% boys?
100%. Every single boy is going to be colorblind because their mother can only give has only small R's to give, and that's where a boy gets his X chromosome from his mom. So I have my mother's X chromosome. So if she gave me a recessive allele, I'm going to express it. because there's nothing to counterbalance it on the Y. So this is why sex-linked conditions, recessive sex-linked conditions are much more common in males than in females, because they only need one small letter and they will express it. So that's true for color blindness and hemophilia, are two human recessive X-linked conditions. What about the girls? What percent of the girls would be colorblind?
0%.
0%, because they're all getting a big R from their father. So they will all have a big R. They'll be carriers, but because they get a small R from the mom, but they're going to have normal vision. This is the usual situation. Recessive sex-linked conditions are passed from mother to son. Okay, but it could ask, do recessive sex-linked characteristics behave differently in males and females? In other words, a man who's colorblind is any different from a female that's colorblind?
There would be no difference, but it would be a difference in the way that they're inherited.
Yes, so you got to remember, a lot of people get just confused. It's just sex linkages, they're inherited differently, but they work exactly the same way in males and females. Genes, that was part of the lab also on genetic simulations. Doesn't matter whether you're male or female, the way this gene works, it doesn't. They work the same way, both autosomal loci, which are, autosomal means they're on the other chromosomes, they're not on a sex chromosome. And sex-linked, they work exactly the same way. And it doesn't matter which parent you get it from or what your gender is, they work the same way, right? That's good. All right. Now, you won't be, like number 11, you won't be asked to draw a pedigree, but if you see a pedigree, remember you should be able to tell a couple of things from it. One, whether it's dominant or recessive. And remember, the way you tell that is if two parents don't express it and they have a child that expresses it, expresses this thing, it's got to be recessive. Right? So you can tell that. If one of the parents has the condition and the other parent doesn't have the condition, and they have children with the condition, you can't tell whether it's dominant or recessive. But if neither one has it and they have an offspring that has it, it's got to be recessive. And then also, if you look at a big pedigree, you can get an idea of whether it's autosomal or sex-linked. If the people who express it in the pedigree, half of them are male and half of them are female, then it's probably autosomal. If the majority of the individuals that express it are male, then you think, ooh, this could be sex-linked, because remember, more males express recessive sex-linked condition than females. Okay. Now, there's a couple other mononym and Mendelian things you gotta look for. Okay, this has been, that corn thing has been Mendelian. Okay, here, number 15. One day you find a blue rose growing in your red rose garden. You decide to mate these two varieties, and to your surprise, you get all purple roses. When you mate the purple roses with themselves, 1/4 of the offspring are red, 1/4 are blue, the rest are purple. What kind of inheritance is being demonstrated? And what is the genotype of the purple roses? Okay, this is a non-Mendelian trait. So, and here's how you determine this. You have a blue rose, you made a blue rose with a red rose, and you get a purple rose. You get a third phenotype. Remember, in Mendelian loci, there's only two phenotypes, dominant and recessive, right? What if you get a third phenotype? Well, this is called incomplete dominance. So there isn't a dominant allele in this situation. Both are partially dominant, so they're both partially expressed. It's also called blended inheritance, because the offspring is like halfway between the two parents. It's like a paint. You mix blue paint and red paint, what color paint do you get?
Purple.
Purple. Just like in the book, it gives you red flowers and white flowers. So you mix a red flower plant and a white flower plant, you get pink plants, because it's blending. It's halfway. So if you have a parent that's 6 feet tall and another parent that's 5 feet tall, all the kids will be 5.5 feet tall. It's right halfway between the two parents. So that's how you tell. There's a third phenotype. The heterozygote has a different phenotype than the two homozygous individuals, right? So the heterozygotes have a different phenotype, and they're halfway between the two parents. And then when you see that on the exam, that will be incomplete dominance, right? That clear?
Yes.
Okay. Now, let me stop screen sharing here. Okay. There's a few other non-Mendelian traits you got to look for, and there weren't any examples of these in this sheets that I gave you. And what are they? Okay, one is called pleiotropy. Remember what pleiotropy is? Have you heard of this? Let me show you where it is in the book. Hang on a second. $160. Remember, so on page 158 is incomplete dominance. On page 159 is multiple alleles. And here is pleiotropy and sickle cell disease. Pleiotropy. Remember, the Mendelian characteristic is that one gene controls one characteristic. All right? Pleiotropy is one gene controls more than one characteristic. Okay? So sickle cell disease exhibits pleiotropy. That is, the first trait is that it causes red blood cells to become sickle-shaped in a low oxygen environment. That's the one trait. The second trait is resistance to malaria. If you have this allele, you cannot get sick with malaria. You see? So we understand that. So if you see something, a problem, and you have one gene controlling more than one trait, you think pleiotropy. Okay? You understand that?
Yes.
Yeah.
Good. Okay. The other, but then you could have the opposite situation. It's also non-Mendelian. And that is one trait controlled by many genes. And that's on the same page called polygenic inheritance. And a way to figure out polygenic inheritance is, if something's polygenically inherited, it has many, many phenotypes. I mean, huge number of phenotypes. So many phenotypes that it fits a bell-shaped curve or a normal distribution, which is on the bottom of page 160 to the left. And so something like height, human height, is polygenically inherited. Because how many different phenotypes are there? I mean, huge, all the way from very, very short. Well, how short can you be and still be a normal height? Somewhere around probably like 4'10" or 4'8" or something. You know, less than that would be condition called dwarfism. And that's a different thing. That's not a normal height. And then about 7'8" or 7'9" is like the tallest normal height, you know, and all those in between. So that's polygenic inheritance, okay? So understand that. You see a normal distribution, think polygenic. All right. And then one more, epigenetics. What does epigenetics means? The role of environment. So you are, all of your characteristics are not just due to the set of alleles you got from your parents. This is the old nature and nurture controversy. How much of you is genetically hardwired, and how much is due to the situations you've been in, the experiences you've had, what you've been exposed to? That will affect gene expression. So even identical twins are going to be somewhat different in the way they approach things, because even if they were like together all the time, they're still going to have somewhat slightly different experiences and things different happen to them. And so they could express those genes differently, even though they're identical twins. See what I mean? I'll give you an example. Human height. It's polygenically inherited, but it's also can be affected by epigenetics, right? It's hard, how tall you are is hardwired. I mean, it's depending upon the set of alleles you have that determine that. But that's assuming you have good nutrition as a child, right? What if you have poor nutrition as a child? Will you reach that genetically determined height? Will you?
Probably not.
Probably not. You'll be shorter because he had poor nutrition. So this is this is called epigenetics. So any kind of a difference between two people due to the environment. And there was an example in the other kind of a book where they have, I think, azaleas. And azaleas can have a red flower or a white flower. It depends upon the nature of the soil they're growing in. If it's acidic soil, they get a red flower. If it's basic soil, they get a white flower. That's epigenetics also. So if you see a problem where the environment is affecting genes, then you think epigenetics.
When is exam 4?
It's going to be I'm probably going to make it available on Wednesday.
And then do the following, like next week?
And then do it the following Wednesday. So I usually
Okay.
open them for a week.
Okay.
And that's going to be on chapters 8 and 9. So it's going to be cell reproduction too. It's going to be mitosis and meiosis too. And a little bit about sex. uh, like fertilization, making gametes, haploid gametes to get together in fertilization, how they get together. I'm going to use a term for sexual intercourse called coitus. That's the official word for, how do people put gametes together? Coitus. They have sexual intercourse, and then fertilization, and then mitosis to become, an embryo, a fetus, and a multicellular, organism. So it has a little bit about sex and the advantages of sex versus asexual. Remember, the chapter started off talking about asexual reproduction. Remember things called parthenogenesis, which is really virgin birth. They're all females. They give birth to diploid offspring that are all clones of each other, and they're all genetically identical, and they're genetically identical to each other and to their mother. And so it also talks about other kinds of asexual reproduction, like fragmentation. Like you can cut off an arm out of one starfish. The starfish regrows the arm, and the arm regrows the rest of the starfish. Well, like in plants, some plants you cut off a branch, you stick it in the ground, the cut end of the branch grows roots, and you get a whole new individual. So that's kind of asexual reproduction also. So all this terminology should be in the topics list for exam 4 too.
Okay.
So so that's 8 and 9 now. I think that's all. Oh, and I'm thinking about the final exam is really going to be on evolution, natural selection. So I'm thinking about what to do about chapter 10. It doesn't really fit into this, the structure and function of DNA. It doesn't really fit into, I don't have it on this exam, and I'm not going to probably put it on the final exam either. So what I'm thinking about doing is making chapter 10 extra credit. Some people have asked for extra credit stuff, and I'm thinking about making chapter 10 extra credit. That is an extra credit exam. Make an exam dealing with chapter 10, and it'll be completely voluntary. You want to take it to get some more points, you can take it. What do you think of that?
So what happens if we take it and we don't do a good job?
Oh, well then there's no downside. Even if you do very badly, you still get those points. See, that's the nature of extra credit is you can only get points. You cannot lose points. So
Okay,
even
then
if you
I
do
think
really
it sounds like a
lousy,
good idea.
so even if you do really lousy, you still get some points. You get those points.
Well, then that sounds like a good idea.
I thought people.
I agree.
Most people think that's a good idea, so I'm thinking of making an exam. We call it exam 5, and make that available pretty soon. Also, it's going to be like 25 multiple choice. No, 25 questions, partially multiple choice, partially matching, and it'll be worth 2 points each. So it'll be like a possibility of getting 50 points. 50-point exam. So I'll make that announcement pretty soon. So as soon as I put that together, I'll make that announcement too.
Okay, I know so many people in the class have talked about the Pearson, but I had like totally forgot about it for a couple of weeks. Now, is all of that due at the end of the semester, or that's just like to help us practice?
Well, yeah, it's due. Each one of those assignments is worth 10 points. And they're open all semester, and they're due at the end of the semester. I will look at how many you've done, and give you points depending upon how many you've done and how much you've done. Remember, you only have to do half of each one to get full credit.
Okay.
And they're not. I don't go in there and grade them. They're automatically graded, and it tells you whether you're right or wrong. But they're really good study guides too. That's one of the reasons why I put them together, because it really helps you study the material. But like I said, even if you haven't got to, you can still get to them. Once I open one, I leave it open all semester. So
Thank you for going over these
Oh, yeah,
genetic problems.
yeah, I know, because usually I have regular semesters. I have these group activities where I give you. Well, this is a group activity right here, where I give you practice on them. But this we can't do that this semester. So I want you to really get practice in doing them. So
Yes, it definitely helped.
good. And I'm recording this, so I'm gonna put it up on YouTube also for if you ever want to refer to it again, and for other people who didn't come into the room. Okay, so if there's nothing else, I'll in the meeting, unless you got other questions.
Nope, I'm good.
You good?
Yes, I'm good too.
Okay, good. All right then. I'll so let's see. Have you guys already done the electrophoresis, or
I do it Wednesday.
you do it Wednesday?
Yeah.
Yeah, I think I did mine last week.
That's right. That's what you did. Okay, good. And then the at-home is allergy ID. Right?
Yep.
Okay, good. All right then. See you
All
later.
right, thank
You're welcome.
Bye. Thank you.